Okay, so this is session 3, Cohort Consortium Project Group Updates. I'm Hazel Nichols at the University of North Carolina Gilling School of Global Public Health, and I am co-moderating this session with Dr. Holly Harris, who I actually will just go ahead and introduce right now because she's also giving our first group update. So Holly Harris is an assistant professor of epidemiology at the Fred Hutchinson Cancer Research Center. And today she's going to tell us about the Associate Member Council update. Just in case anyone's joining us newly for this session, I also wanted to say we're going to go through 4 talks in a row, and then we'll save questions for the end. So thank you very much.
Great, thanks Hazel. So I'm going to go through the Associate Member Council update, so we can go to the next slide. And this is on behalf of the Associate Member Representatives, so not just myself. So the first thing we wanted to talk about really is who is the Associate Member Council, because we're relatively new. And so generally we're a representative body of early career investigators within the Cohort Consortium. And membership is really targeted to postdocs, researchers, and early career investigators, those who are within about 10 years of terminal degree, but it's not like a very hard cutoff to be a member. And to connect the group with the Cohort Consortium, we really wanted to be open to anyone who's either identified as part of a research team of one of the participating cohorts, or anyone leading projects that are based on Cohort Consortium data. And so sort of by default, all investigators who meet these criteria are already considered members of the AMC. But membership is voluntary and it's at no cost. The biggest thing for us now is identifying members in their emails so that they can participate in activities. And I'll talk more about that in a bit. And so just some confusion we've had about the difference between the Associate Member Council, which is the broader group, and then the AMC Representatives, which are a smaller group sort of that act as the steering committee of our group. And I'll talk about that in a few slides. And so on the consortium website, you can find the AMC's charter with details on membership and a variety of other things if people are interested in those details. So next slide. So what's the purpose of the AMC? So it was started following the 2018 annual meeting under the great leadership of Celine Vachon, who really spearheaded and got this going and brought us together. And it's part of the career development portion of our consortium. Over engaged board investigators, and we do that back to network to research collaborations within the consortia. And the idea is really to position members of the next generation to eventually take on leadership roles within the consortium. So we focus on communication, education, training, and collaboration in the work that we do. Next slide. So the AMC representatives are sort of we call it the steering committee for early career investigators that helps set the priorities for the AMC's activities and actually to get the work done and to plan the events. So it consists of early career investigators who should represent the demographic, geographic, and scientific diversity of the AMC. We meet usually monthly to discuss and plan events that surround the AMC's purpose. And I'll talk more about those events on the next slide. So the idea is that the representatives will serve up to a 3-year term. Because we're so new, we haven't actually reached that yet. But when the time comes, we'll have elections where people can be nominated to be an AMC representative. And we'll probably try to time those elections with the broader steering committee elections. And there's more information about the nomination process in our charter that's on the website. And then we also connect with the steering committee through both our steering committee liaison, who was originally Celine Vachon, and then this year Shine Chang has taken on that role. So they're really great mentors to our group to help make sure that what we're doing is in alignment with what the steering committee is doing. And then we have one of our representatives sit in on the steering committee monthly meetings to again make sure that we're all on the same page with the activities we're planning. So next slide. So activities that we've done in the past 2 years. First, we created a charter to sort of give us some guidance around our activities. Then in 2019, we had a kickoff session at the annual meeting, and it included a roundtable event where we had 2 senior investigators at each table and an AMC representative. And all the participants rotated from table to table, discussing different topics that included getting started on consortium studies, how to lead projects and papers within the consortium, and how to facilitate career development within the context of the consortium. And that activity was really well received, and we got a lot of feedback from it that's going to help us plan future events. Then in the past year, we've been really working on compiling an email list for the AMC, which is a little bit harder than it seems. We have initial emails from people who participated in our kickoff event. We solicited names from cohort leadership of junior investigators that they thought would want to be a part of the group. And then as we've had different events, people will email and ask to be on our email list. So if you know people who maybe aren't on the email list, feel free to connect with the consortia leadership so that we can include everyone. And I'll have an email that people can contact on a later slide. And then most recently, we put together a webinar on supporting early career investigators in cancer, sort of during this pandemic that we're all living through. And there were over 120 attendees that saw the panel presentations that was presented. And we also got feedback after that, which will help plan our future events. So next slide. So for our future plans, we'll probably do more career development webinars, including grant writing. People have suggested doing a webinar and elevator pitch, so we're going to work on those ideas. And we really want to grow our AMC email list. We have about 80 people on it right now, but we want to make sure that everyone who's interested in our activities can be informed of what's going on. And so you can email the email that's on this slide to get on the email list if you're not receiving our emails already. And I know we're saving questions for the end, so thank you.
Great, thank you. Our next speaker is Dr. Jill Koshiol, the senior investigator in the Infections and Immunoepidemiology branch in the Division of Cancer Epidemiology and Genetics at NCI. And she's going to talk about updates on the biliary tract cancers pooling project.
Jill, I think
Are you on mute possibly?
Jill, if for some reason your computer microphone isn't working, you're able to change your audio connection by going to the mute icon found in the lower left-hand corner of your screen and clicking the up arrow, and you can change your microphone or your audio settings. And if you are calling in from a phone as well, make sure you're unmuted from the phone. Hazel, I don't know if we want to jump to the next presentation and then come back to Jill when she joins, but it looks like she's just calling in the next few minutes.
I'm happy to go ahead. I think I'm next.
Okay, great. Well, why don't I then introduce Dr. Peter Campbell, the Scientific Director of Population Science at the American Cancer Society, who will update us on the liver cancer pooling project.
I'll just wait for my slides. Thank you.
Sure.
Okay, so good afternoon or evening or morning, everybody. It's really my pleasure to update this, or to give this update on behalf of the Liver Cancer Pooling Project. As shown here, Catherine McGlynn at NCI is the PI, and she's really a world leader in this area. It's been just an absolute honor to work with her these last 10 years or so. And it's entirely in line with her character that she asked me to present today and share some of the spotlight that she and her team really deserve. Next slide, please. So when we talk about liver cancer, we're really talking about hepatocellular carcinomas and intrahepatic cholangiocarcinomas in particular. So the split here is about 70% for HCCs and about 15% for ICCs. The rest is really quite rare. And when we launched this project, again, 10 years ago, there had been rising rates and incidence and mortality since the late 70s or the early 80s. I think it was like a threefold increased risk or increased rates over that period of time in the US. So there were hypotheses to explain those trends, but there wasn't a lot of empirical data, and that was a large part of the motivation here. Now, an additional motivator is the fact that the disease is still pretty rare. And, even with these rising trends, rates are under about 10 cases or so per 100,000 people in the US. So the rarity, the fatality, those factors both made it really conducive to a Cohort Consortium project. And then after a lot of discussion, we intentionally chose to focus on North American-based cohorts. And that was to avoid any potential etiologic heterogeneity, primarily by hepatitis or hep C viruses, with the underlying causes from other countries. Next slide, please. So we're really fortunate to collaborate with 19 prospective cohorts. They're all listed here. All are from the US except for one Canadian study. And as you can see, really outside of AARP, it's pretty clear that no single study could really do a whole lot on their own, in terms of publishing, a scientifically rigorous manuscript on liver cancer etiology. So for most analyses, we have about 1.2 million total study participants and about 3,000 cases. And we have almost 800 cases with a pre-diagnostic blood sample. So that's just, in this area, it's just an absolutely unparalleled area for biomarker discovery. Next slide, please. So we've published 9 key papers from this endeavor. The topics range from excess body weight in several forms to aspirin use, coffee intake, alcohol and smoking, and reproductive factors. We've also published 1 key biomarker paper on sex steroids in women. That's shown on the right in the middle. And there the hypothesis was that estrogenic hormones might explain some of the lower liver cancer risks that we see in women compared to men. But curiously, that didn't pan out. And then last, Catherine is working on a similar sex steroid paper in men that we hope to see hopefully soon or early in the new year. And next slide, please. So switching now to one of the ongoing projects, this is a project, that as a person who thinks a little bit more about colorectal cancer etiology, it's really quite close to my heart. So this idea of a gut-liver axis relates to how the liver and bowel communicate through the biliary tract, the portal vein, and through some biochemical messengers in circulation. So there's a lot of crosstalk back and forth between those organs. And because of high throughput technologies, we're starting to learn a little bit about how GI tract diseases really form sort of a continuum. They're not simply stochastic. So we can measure markers of gut health, and some of the really strong ones include antibodies to lipid polysaccharides and flagellin. When we find those in circulation, we know that they shouldn't be there. They escape through the luminal wall, through the colon wall. So it's an indicator of a leaky gut. We can also measure things like bile acids, which are measures of liver health and function, and then some of the other biomarkers that we see here. And next slide, please. So you saw Jessica's name as first author on, I think, 6 of 9 of those papers we saw a few slides ago. Jess did her postdoc work with Catherine at NCI, and BU was incredibly fortunate to scoop her up, I think a little bit over a year ago, if I remember correctly. She's emerging as one of the true leaders in this field, and not to mention just a really talented and wonderful person to work with. So in the context of this project, Jess is leading it, and we plan to start running the assays as soon as the remaining samples arrive at NCI, and I believe we're very close to arriving at that point. Next slide, please. Now, currently, we're also doing exploratory work in just a handful of the LCPP cohorts. So we plan to bring these efforts to the whole project as soon as we have some promising data, we hope. I'll touch on each of those now just briefly. So next slide, please. So one of the investigators at NCI, Jing Wang, he identified, or his group identified, a viral exposure signature for hepatocellular carcinoma. This was published just this summer in Cell. And if you're interested at all in risk prediction, translational omics, or early detection biomarkers, I strongly encourage you to seek this paper out, even if the outcome isn't necessarily your area of expertise. No, it's just it's a really compelling read. Next slide, please. And the gestalt of the paper went a little bit like this. So the VIRScan technology identifies exposure to known human viruses. Jing's work found an HCC signature that was comprised of 61 viral strains. Some were positively associated with risk and others were negatively associated with risk. So the major positive association was with HCV, which is not surprising. It actually works as a good internal control. But the AUCs for this scan were over 90%, which is really quite amazing for an early detection panel for a disease that is fatal and really has no screening modalities. And next slide, please. So Catherine and colleagues are first planning to do a confirmatory study in PLCO to show that this technology is amenable to frozen samples and to the sorts of data that we're accustomed to working with. So PLCO is also a good starting place because they have HBV and HCV status. So we have another type of internal control for both cases and controls. And PLCO also has a GWAS completed for most of those cases and controls. And the GWAS data were important here. Again, you should go back to the paper if you're really interested in it. But there was basically a susceptibility locus embedded in part of the story with the viral outcomes. And next slide, please. So Hong Zhang, he is the fourth member of our virtual relay team in LCPP. Hong is at Harvard. And much like Jessica, he's one of those really incredibly talented and hardworking junior investigators. He's also a wonderful person to work with an incredible work ethic. And as a testament to that, he has 3 R01s that are listed here that are under different stages of review. Just on Friday, we saw the pink sheets for the PFAS and liver cancer grant. There's an awful lot of positive comments on its significance and innovation. PFASs are a group of chemicals that we find in consumer products like cookware, food packaging, stain repellents. So those chemicals target the liver where they're metabolized, and at least some animal and some heavily exposed occupational data suggest that those chemicals are also associated with liver cancer. So then for a second project, we're starting to learn that Helicobacter pylori has effects that go beyond the stomach. We've known that it's a class 1 carcinogen in the stomach for decades. Helicobacter species, they can colonize in the human liver or in the tissue of the human liver, but we don't really know if those exposures are associated with liver cancer risk. So that the objective of the second proposal from Hong is to assess associations between H. pylori related species and liver cancer risk. And then third, there's emerging evidence that high magnesium intake is associated with a reduced risk of hepatocellular carcinoma and mortality from liver diseases. So this proposal is to test a hypothesis that magnesium modifiable metabolite signatures in bile acids and are associated with lower risk of developing hepatocellular carcinoma. And then last slide, please. And I'll just wrap up here and thank you for your attention. And I've asked to make one plug that if any additional cohorts outside of North America are interested in joining LCPP, to please reach out to me and Catherine. And in terms of future directions, we also hope to add dietary studies to the portfolio shortly. And thank you again for your attention.
Great. I think we're going to actually try to go back to our second talk on the biliary tract cancer pooling project, if Dr. Koshiol has audio now.
Testing 1, 2.
Yes, we hear you.
Victory. Okay. So once again, sorry for the technical difficulties, but hopefully you can hear me well now. Thank you again to the organizers for giving me this opportunity. Next slide. So we had our working group meeting at the same time as the liver cancer pooling project on October 28th with over 30 participants. So it was great to see so many people interested in these rare cancers, which I think are really the ideal of one of the ways that the Cohort Consortium is extraordinarily valuable and useful to be able to allow us to study such rare cancers. So in this working group, there were several presentations, both by myself and by Dr. Sarah Jackson of NCI, Dr. Lona Ajirian of NCI, and also Dr. Wei Zhang of Vanderbilt University, who presented a proposal to evaluate metabolomics in a consortium that he's actually pulled together outside of BitCap, but that we hope to be able to collaborate on and so that we can synergize our efforts. And that led to quite a bit of really valuable discussion, both on our ongoing work and our future study plans. And there was particular interest for studies that involved biospecimens. Next slide, please. Just to give you a sense of the scope of BitCap, here you can see the cohorts that are contributing, and I think the bottom of the slide might be cut off just a little bit, but it just gives you kind of an idea, along with the cases and the non-cases that they contribute. We currently have 29 prospective studies involved, and we're working on bringing in the California teacher study, which will bring us up to 30. We have 2.9 million participants and over 5,000 incident biliary tract cancer cases. Next slide, please. For those studies that are already within BitCap, here you can see the distribution of cancer across anatomical sites. We have over 1,700 gallbladder cancer cases, over 1,500 intrahepatic bile duct cancer cases, over 1,400 extrahepatic bile duct cancer cases, and nearly 800 ampulla of otter cancer cases. You can also see the sex distribution. As expected, we have more female than male gallbladder cancer cases, and there's a tendency towards a male predominance for the other types of biliary tract cancers. But keep in mind that, of course, this is heavily influenced by the composition of the studies that contribute, and some of these studies are male only or female only. So this is not a true distribution of the sex ratio within biliary tract cancers, but it gives you the picture of what we have. The median age at diagnosis is 72 to 73 years of age. Next slide, please. Looking at our participants overall, the median age is 58 years, with 60% being female, and the vast majority, almost 80%, being white. We do have a pretty substantial proportion of Asians and Pacific Islanders as well. Next slide, please. We've been successful in publishing a number of studies that I think are really important for elucidating our understanding of the etiology of biliary tract cancers. And that's valuable because prior to BitCap, there was really very little evidence in the literature giving us prospective data on risk factors for cancer across the biliary tract. Most prospective studies that did exist were pretty small and typically produced conflicting evidence. So even for classic cancer risk factors like smoking and alcohol, we really didn't have a good handle on how they related to cancer within the biliary tract. And that's just one example of how BitCap is helping us to move the field forward. Next slide, please. Here you can see a summary of the analyses, the database analyses that we have ongoing and planned for the future. We're currently working on an analysis of exogenous hormone use, including oral contraceptives and menopausal hormone therapy, as well as a study of diabetes. And you can see the cohorts that contribute listed here. Now, to kind of follow along what Pete was saying, if you don't see your cohort listed here and you're interested in participating, by all means, please do follow up with us. We'd love to work with you and get you involved in these analyses. You can also see our future studies. We're planning to work on an analysis of aspirin and NSAID use and also statin use. Again, you can see the cohorts participating listed here. And we know that there are a number of cohorts that have data on these variables. We just don't have those data in hand. So again, if this is something that's of interest to you, by all means, please do follow up with us. Next slide, please. Just to give you a little bit more impetus for the statins analysis in particular, here I'm showing you some data from a study that we conducted within the UK Clinical Practice Research Data Link, looking at statin use and risk of developing biliary tract cancers. And as you can see, what we found was that current statin users had a reduced risk of developing biliary tract cancer. And that reduction in risk was most pronounced for long-term users, as indicated by the number of prescriptions and increasing cumulative dose. But even with 3,000 biliary tract cancer cases in this study, we didn't have the power to really look at each anatomic site within the biliary tract, which we know is important. And that's something that we would really like to be able to do within BitCap. So it would be just fantastic if we could get some additional cohorts involved in this analysis. And the findings will be important because if we continue to see this inverse association, it might suggest that statins could potentially be used as chemopreventive agents, particularly in high-risk populations, such as patients who have primary sclerosing cholangitis. Next slide, please. So finally, I'd like to wrap up by describing a proposal by my postdoctoral fellow, Sarah Jackson, to evaluate circulating hormones and biliary tract cancer risk. Now, Sarah is especially interested in how sex contributes to the development of cancer and why it's different at different cancer sites. And we think that biliary tract cancers are just a great place to address this question because there are very different profiles in the sex distribution for these cancers. For example, gallbladder cancer is one of the few cancers that's more common in women than in men, with about a 2-to-1 female-to-male predominance worldwide. But most other biliary tract cancers have a male predominance. And the gallbladder is sensitive to changes in hormonal concentrations as it expresses both estrogen and progesterone receptors. And high circulating levels of female hormones have been associated with gallstones, which is the single most important risk factor for gallbladder cancer that we know of. Now, stones do contribute to the other biliary tract cancers, but not to the extent to which they contribute to gallbladder cancer. In BitCap, we found that parity was associated with increased risk of gallbladder cancer and intrahepatic bile duct cancer, but not with extrahepatic bile duct cancer or ampulla of Vater cancer. And in the liver cancer pooling project, Dr. Jessica Petrick led an analysis of circulating hormones and found that higher levels of circulating estradiol was associated with increased risk of intrahepatic bile duct cancer. So we think an important next step to better understand the biology of biliary tract cancers is to evaluate circulating hormone levels and gallbladder and extrahepatic bile duct cancer. Next slide, please. Therefore, we propose to evaluate the associations between sex steroid hormones and levels of gallbladder cancer and extrahepatic bile duct cancer risk in postmenopausal women and men aged 50 and above. We hypothesized that increasing levels of estrogen will be associated with increased risk of gallbladder cancer, while increased levels of androgens will be associated with increased risk of extrahepatic bile duct cancer. Next slide, please. Based on our power calculations, we think we need about 111 to 150 or so cancers for both gallbladder cancer and extrahepatic bile duct cancer in both women and in men. And so we've listed here some cohorts that could potentially contribute to this effort. Now here we're focusing on non-Asian cohorts because we found in our database analysis of reproductive factors that the associations were different when we looked at Asian and non-Asian cohorts. So as a place to start, we thought we would begin here. And the assay does require 0.4 mils of plasma or serum, which we recognize is a substantial amount. So of course, we'll be working closely with the cohorts to evaluate both their interest and the feasibility of being involved in this effort. Next slide, please. So just to wrap everything up, BitCap is providing important new insights into biliary tract cancer etiology. We have several database analyses that are ongoing or planned. We do think it's important to take the next step to digging into the biology by evaluating biospecimens. I think that Sarah's proposal to look at circulating sex hormones is a great example of that progression. And I'm also really pleased to see Dr. Wei Zhang working on the metabolomics to understand the biology. And I highly encourage you to follow up with him if you're interested in that approach. And with that, I will thank you for your time and attention.
Great, thank you very much. I think we're going to move on to our fourth and final presentation for this session from the Ovarian Cancer Cohort Consortium, or OC3. And we'll have two speakers today, Dr. Renee Fortner from the German Cancer Research Center and Dr. Britton Trabert from the National Cancer Institute.
All right, can you hear me?
Yes, we can.
Okay, perfect. Good afternoon, good evening, good morning, everyone. It's my pleasure, together with Britton Trabert, to present some of the information on new initiatives and future directions in somewhat of a broad overview kind of format that we have ongoing in the Ovarian Cancer Cohort Consortium, or the OC3. Next slide, please. So first of all, certainly I know that many of you are familiar with our consortium in kind of broad terms, but I thought it would be useful just to give a very brief history, so you can understand where we're coming from when we talk a little bit more about where we're headed. So first, we are a consortium that was originally funded by the DoD, and we were first established in 2012. Shelley Two-Roger, who was at that time at the Brigham and Harvard Medical School, was the lead PI, and she established the Data Coordinating Center there, which was really instrumental in all of the gathering and harmonization of the baseline data and enabling some of our early investigations. So here we're referring to kind of our earlier phase that we are now just kind of moving out of as OC3 version 1.0. And we have some of the highlights from that phase listed here. So as I mentioned, we had established a data coordinating center. We had all of the baseline data for initially 21 prospective cohorts, now 27, harmonized at Harvard. And we did quite a number of interesting investigations looking at these baseline data with respect to known and putative risk factors, but also some more novel risk factors, and looking at ovarian cancer risk by histotype and by anatomic site. So ovarian cancer is not a very common cancer at all and is quite heterogeneous. And so the OC3 was really key in allowing us to understand some of these associations well in a large prospective cohort consortium setting. We also moved just beyond questionnaire data and worked on the pooling of existing biomarker data. So in these studies, we took data from existing nested case control studies within our participating cohorts, centrally harmonized that, and did the analyses to, again, really allow for the first studies that had the statistical power to evaluate ovarian cancer associations by subtype. Of course, this led to lots of great ideas about using de novo biomarker assays and studies within the consortium, but that we have saved kind of for our next phase. Nico Van Sensen was key in establishing a genetics database to understand
It sounds like we lost her. Britton, are you
Yeah, sure.
able to jump in for?
We lose her completely. Yeah, so to follow on what Renee was saying, so Nico was led the establishment of a genetics database, and that is just getting to the point where we will be able to use that. And as a result of kind of this first wave of OC3, we've published 10 papers to date, and we have a baseline paper that is currently in progress, really describing kind of the effort and the data that exists and the possibilities for the future. I think one of the really
Britton,
important—
I'm back.
Okay, go ahead, Renee.
Sorry.
No problem.
You're doing fantastic pinch hitting there. Okay, so I just wanted to finish off this slide and say that I think one of the really fantastic things about the OC3 for myself and for, I don't want to speak for others, but I think also for others, is that it's a very active group, and we have many different levels of investigators really across the career continuum. where investigators have been able to lead studies from a more junior level all the way through quite senior investigators. Okay, next slide, please. So now, OC3 version 2.0, and I think this is quite an exciting phase for many of us. So we have had really quite a lot of changes in the last years. Britton will talk more about some of this as we move forward, but I'll hit on just some of the highlights in terms of some of the extension and expansion that we've had in the consortium through some recently funded projects. So first in this is that we're moving now beyond just the baseline data. So we're in the process of collecting and harmonizing follow-up data for the cohorts that have such data, as well as gathering some of the baseline variables that potentially we wish we had collected in the first round of data collection. And this is largely thanks to Britton's new DoD-funded study on aspirin use and ovarian cancer risk within the OC3. Another exciting aspect is now the expansion of racial and ethnic diversity within the OC3 via NCI supplemental funding, which was awarded to Melissa Merritt. And this will really expand the consortium to make it a much more diverse set of cases and underlying cohorts. And then finally, there is a adjacent consortium called PREDICT. This is also funded by the DoD, and this is specifically looking at early detection biomarkers for ovarian cancer among the subset of cohorts that have existing biospecimen resources. As I mentioned on the previous slide, hopefully before I dropped off, we have done some pooled studies using existing biomarker data to date. And now we're really looking at conducting studies in a much more collaborative way using samples from the cohorts themselves. So we have studies that are ongoing as well as studies that are planned using both blood-based markers, so using, for example, serology for infection-based markers, as well as using tissue markers with extended tissue collection and new staining of existing tissue microarrays. And with the next slide, I'll hand it off to Britton.
Thanks, Renee. So in terms of kind of this moving forward in terms of OC3, Shelley to Roger moved to the Moffitt Cancer Center. And so with that, the Data Coordinating Center moved with her. And this really gave us an opportunity to rethink data infrastructure and harmonization. And then that dovetailed nicely into the opportunity to collect follow-up data. So if you could go to the next slide, please. So this is a kind of schematic, which I'll describe first, that is from our descriptive paper that we're putting together right now that really highlights the timelines of the individual cohorts that have contributed data. Each line represents a cohort. The triangles represent the data collection time points, so the questionnaires, and then the red circles represent the initial enrollment period and really represent the baseline data that we have collected to date. So if you go to the next slide, you can see the summary of the 25 or so cohorts that have been included in our initial papers in some way, shape, or form. And as you can see with the triangles, there are over 100 data collection points where we're going to be collecting additional information to do analyses that will update exposure information in terms of looking at ovarian cancer risk. Next slide. And so as part of this, with the effort at Moffitt, we are working closely with the biostatistics and bioinformatics shared resource. And this is the data harmonization process, a schematic of the data harmonization process as it is being currently implemented. And so I'll kind of walk you through this and we'll follow the numbers on the slide. So in the top left, we have the individual, at number one, we have the individual cohorts that are contributing updated follow-up data and updated baseline information. And those are getting uploaded directly to a Moffitt's share file system. Once it's in the share file, it's downloaded by Research IT behind the Moffitt firewall to a local folder where our Moffitt OC3 team that includes a number of people from the biostatistics and bioinformatics shared resource downloads that raw data set to Opal where the data harmonization is done. It's then an iterative process back and forth in terms of defining what the end product of the data looks, what the end product of the variable looks like, and what the individual cohort has collected. And we're kind of in the early stages of working the kinks out in that project. Once the harmonization for a study is completed, data set can be, the harmonized data set is uploaded back to the share file. At that point, which we don't have depicted on the slide, we can send that harmonized data back to the cohort consortium and resolve any, the specific cohort and resolve any issues, and also share that data back with them. But importantly, it can be downloaded to SAS, which can be accessed by both individuals internal to Moffitt and the kind of large expanse of investigators working on the Ovarian Cancer Cohort Consortium and other institutions. And so the primary data analysis will really be done, it will be conducted in this in this SAS research infrastructure. What our future plans are in terms of data harmonization are to also link the Opal system with the MICA data exploration tool that is available. And so that once things are harmonized, the plan there is to share, link and allow data exploration through a web-based search engine that really allows an individual to either within the Moffitt system or on a website to browse, query, and compare study and metadata. And so in terms of moving forward, we'll go ahead and go to the next slide. We just really wanted to emphasize that it takes a team. As Renee said, we really are supporting kind of a cross collaboration of investigators from many levels. From the perspective of the co-chairs, we would like to thank the individual cohorts that have contributed to our effort thus far, and we're happy to welcome additional cohorts that have ovarian cancer outcomes to be included. We have an active steering committee that we're continually adding kind of new investigators to. I want to specifically give a big shout out to the data harmonization team at Moffitt, and so that is led by Brooke Fridley. Includes Wen Yi Feng, Rodrigo Carvala, and Jessica Martinez Monta. And so this is within, again, I mentioned it before, the biostatistics and bioinformatics shared resource. Our contact, if you want to reach out to us, is Jessica Martinez Monta, and this is her email. We also have a website you can visit, the oc3.org. Her email is on there, and we've listed our funding here. If you'll go to the next slide. Next slide, please. Oh, there we go. We just wanted to say thank you to everyone. This is a picture from our poster last year. I'll point out on the far right is Mary Townsend. She's our project coordinator at Moffitt and has been critical to this effort. We owe Mary a huge debt of thanks in terms of coordinating and getting everything moved to Moffitt, all of the data use agreements and everything in place. And then between Shelley and Renee in the middle there, that's Jessica. She is also critical to our endeavor and deserves a big thank you. And so with that, we'll say thanks.
Great, thank you. I think we'll open up to questions now, which you can put in the chat. And I will read the first one, which is for Britton, about how the data sharing agreements were established with OC3. So you've developed a very enviable data sharing infrastructure. From version 1.0 to version 2.0, did you renew the DUAs with more freedom, or did you have that amount of foresight in 2012?
No, so with the transition of the data coordinating center from the Brigham to Moffitt, it required us renewing the DUAs with all of the cohorts that we had existing DUAs with. And so when we did this renewal, we gave the cohorts really two options. They could we could do a three-way agreement between the Brigham, Moffitt, and the cohort, or we could establish a new agreement with the cohort and Moffitt. And so each different cohort kind of picked their own path, but we did redo all of the data sharing agreements with the move from the Brigham to Moffitt. What we did as part of that data sharing and the update was to expand to be able to include the updated follow-up information, and then also any of the bio specimen kind of information that we thought would be useful in terms of if people had existing TMAs, et cetera. So great question.
Thanks, Britton. So the next question we have, which I think applies across all of the working group updates, was could the speakers address sort of how you access the data for all these projects? How can people apply to access the data? Do you allow people from outside your group to use the data? And so why don't we start with Jill and then go to Peter and then end with Britton and Renee.
Okay, well, we actually haven't had anybody that's been particularly interested in accessing the data yet. And so it's a little bit of it would take some working through. I think it would be possible, but we would certainly need to talk with all of the cohorts because of the data use agreements that we have. So we have the data at a sort of an honest broker site, and all of our analyses are done within NCI. I think to share those data, we would probably need to update those agreements to allow that to happen. It depends a little bit on what you want to do. So for example, Dr. Wei Zhang's effort, the way that we're trying to synergize with that in part is by sharing our harmonization schemes so that we don't have to reinvent the wheel. That way we may not be able to share the actual data, but we can at least say this is how we coded things and kind of go from there. But I'm interested to hear what others' approaches are on that.
Yeah, and for the liver cancer project, it's the same sort of situation. We haven't been approached by external people. We would certainly be happy to, but much like Jill said, I think if an outside investigator was really interested in running the data herself or himself, it might be more DUA and MTA problems than it's worth. I think what might be more realistic is if an external investigator had a research question and we could run the data from NCI through that honest broker database and then provide summary tables based on an analysis. I think that would be completely feasible upon appropriate approvals and whatnot.
Sure. So in terms of OC3, I'd recommend going to the OC3 website. We have a proposal system that follows the Cohort Consortium proposal, but we really kind of review those and look to kind of heavily collaborate with investigators interested in utilizing the OC3 data. As I pointed out in our presentation, we will be able to grant access to the data through a SaaS system, but we ask that individuals have an approved proposal to be able to get access to that. In terms of the individual cohorts that contribute, if there is existing data from a cohort, we have kind of the process that is outlined in the DUA is that it's an opt-in, and so we send for each project that we're working on, we send an email to the individual cohort lead to have them opt in to have their data used for that project so that there's a completely kind of transparent process in place for working on projects.
Great, thank you. And so we have another question for you, Britton, about how would the DUA DTA process work for new cohorts that join?
Sure. So for new cohorts that are interested in contributing data to OC3, we'd set up a DUA or a DTA with Moffitt, have the cohort set it up with Moffitt directly for sending data to the data coordinating center within the same construct that we have in terms of for the other cohorts.
Great, thank you. And so I have a question I guess for all of the working groups. So are there any keys to engaging early career researchers? I know that both Peter and Britton specifically talked about postdocs and people that they bring in to really get involved in projects. Are there any like advice or ideas you have behind how to engage them?
Wow. I was an early career investigator when this started, whatever that's worth. Obviously much later in my career now. I think if you build a good resource, the junior investigators will come. And I think when you look at the track record of LCPP and BitCap, which I've also been involved in, and the quality and caliber of postdocs that have worked on those projects, if you build a resource, those outstanding junior researchers will come to you. And I think we've been really successful just because of the resources that Jill and Catherine have built.
Yeah, I would echo what Pete says. I think the ability to kind of advertise and talk about these projects yearly as part of the Cohort Consortium, I think has been key. But it really, Shelley and Nico started this in 2012 and we're very open to including people. And it really has taken a team to get it off the ground. And they continue, they're continuing to, and, honestly, we're continuing to include new people. We have very open meetings in terms of talking about data analysis that really engage everybody on the call. Shelley is coordinated with Katie Terry at Harvard and has really converted it to kind of a large scale every other week meeting that initially started out as kind of the ovarian cancer interest group at Harvard that includes kind of everybody who continues to work on ovarian cancer who came through their system. I participate in that and it's open to including our postdocs and other trainees. And it's really an environment where we can practice presentations, we can review grant aims, and talk about research findings. And so it's really been a great collaborative network that has been set up with some really, in terms of vision going forward. And I think we've developed a really good collegial working relationship, and we're happy to kind of share the resource and have other people who are interested in ovarian cancer be involved.
I would just add for the OC3 and just to a bit highlight some of what Britton said. I think the enthusiasm from above really is infectious to middle and earlier career investigators. And the resource is fantastic, and having interest from early career investigators is fantastic, but really having that strong enthusiasm from the top, I think really made quite a difference in our consortium and getting people not only engaged and excited, but really making progress and putting projects forward and getting their own funding within the OC3.
Thanks. Jill, do you have anything to add to that before we go to the next question?
Sure. I just wanted to hop in and say I kind of echo everybody else. Just having this platform is really useful to share what we're doing, and it is just inordinately clear to me that it's our early investigators that make things happen. What was it, Pete? 6 out of 9 papers that Jessica wrote for LCPP.
Yeah, it was 6 out of 9, and then of the remaining 3, one was Sarah, who's also junior, and then the 8th and 9th were one for me and one for Catherine. So we had to make some contribution, I guess, and that's what it was.
But yeah, I think our early career investigators they're clearly vital for moving this work forward. So having this platform to share that kind of work and get the word out there is great. And I also hear what you're saying, Renee, like when I'm able to share my work at other places, it often gets people interested and excited, and sometimes they'll follow up with me. Sometimes if I know they have an interest, I'll follow up with them. So for example, one thing that I didn't talk about in this session was that Erica Loftfield is very interested in coffee and tea consumption and how that relates to liver cancer. So we're also interested in how that might be relevant for biliary tract cancer. So she and I have been talking a little bit about how that might look in BitCap. But I think that these kinds of forums are really the main way that we can make people aware of what's going on, and then get them involved, because we certainly need the involvement of those early career investigators.
Great, thank you. So another question that I think everyone can see in the chat, probably deserves a much longer discussion, but is a question on how to, besides engaging the early stage investigators, how to increase the pool of investigators that are underrepresented minorities. I think this is true within the consortia and broader. So I don't know if anyone has any thoughts on that.
Someone else, I think, mentioned, I think that was Maria Martinez, wasn't it, that asked that question? I think someone else posed at least a suggestion of having fellowship funding. And that might be, one way to approach it. And I think Elena is trying to offer a question or an answer too.
So we have.
Thank you. There she is.
Thanks, Pete.
I hate speaking for you.
Jim wants to comment, but we, after everything that happened in this country, the CTS and one of their steering committees, we devoted one whole meeting, and a dialogue continues on, even though the CTS is not diverse, what we could offer to diversify the pool of underrepresented minorities. And so we have some plans. I can say there is still a dialogue. We actually put something in our website on diversity and inclusion, expanded that. And we have sort of some metrics that we would like to follow just to keep us in check. So I think that's where we're coming from. And again, this is an early dialogue that I feel needs to happen in every single consortia. So Jim, I don't know if you want to comment.
Just a brief addition. We did talk a lot about, what can we do as a cohort? And there are, obviously we can't change our population, and we're somewhat limited by the scientific ideas that come up. But whenever we think about expanding sort of our partnerships and looking at who's using our data, are there things we can do as active outreach to try to make sure that everyone across the community has access, people who haven't had access before get particular, whatever help and assistance we can provide.
It sounds like this might be a great topic for next year.
Yep. Yeah. Thank you, Elena, for bringing that up. So a question I have for, I guess, OC3, but as well as for the other groups is, you've focused primarily on looking at risk and incidence so far. Are there any plans to move into looking at survival and mortality in future projects and how you might facilitate that?
Sure. I think that's a great question. I mean, we've definitely submitted some proposals for funding in terms of being able to look at survival outcomes for ovarian cancer. One thing we talked about recently at our at the OC3 Cohort Consortium meeting was really figuring out kind of what needs to be, what preliminary data we need for the grant proposals to be able to be successful in getting funding for that. So as part of the kind of collection of follow-up data, if there's additional information, and we are including information on mortality and outcomes to the extent that it's available within cohorts. And our plan is to collect some of the first-line treatment data from the cohorts that have it to really see if we can collect that. It makes sense and looks good. So we are definitely thinking about ovarian cancer survival and kind of seeing if we can transition the resource to be able to answer those questions as well. But I think there's kind of a lot of groundwork that still needs to be done in that space.
Great, thank you. So we're gonna wrap up since we've reached the end and don't wanna cut into the next time, but I think there's more engagement that we can have later on all of these questions. So thank you to everybody that presented.